So I think when I start the recording, are you getting a consent or something? Can somebody tell me that? To yes, say yes or no. Okay, let's proceed. We will discuss this later. Yes, in ribosomes, I think we have learned about several things. In that, there are two subunits. One is the small, and the other one is the large subunit. In prokaryotes, it is small is 30s, and the large one is 50s. And in eukaryotes, small is 40s and the large one is 60s. S is Wordberg units. It's a sedimentation rate, and the larger ones will sediment faster than the smaller ones. And each small subunit and large subunit, they have several proteins. This may have in eukaryotes up to 50 proteins or 30 proteins, and it has one rRNA, that's this 16S rRNA, say, in prokaryotes. And in the large subunit, you have about 50 proteins and two rRNA molecules, 28S and 23S and 5'S and so on. So we learned so far about the different components that are required for translation to occur. We have also learned some important aspects of amino acid, how the tRNAs are charged, and also about the general reaction of peptidyl transfer. So translation is broken down, you can say it as into initiation until here, say for example, the organization of the ribosome on the mRNA. So the mRNA should be recognized and the ribosome should organize like this at the ribosome binding site. Right? In prokaryotes, it's called as Shine-Dalgarno sequence. In eukaryotes, it's called as Kozak sequence. And in today's class, we also will try to study the elongation, where after initiation, how it proceeds, and some of the things are interesting, and we have to learn about that. And eventually, the termination, wherein the end of the open reading frame, that's the stop codon comes, and then how these different things like the protein that is produced, ribosomes, and mRNA are released, and how ribosomes can be recycled and mRNA can be again read. While doing so, we also learned about what is polysome. I hope you remember that. Polysome is mRNA loaded with several ribosomes like this. Okay, so for translation initiation, the ribosome should recognize the mRNA. And it is with the job of the small subunit. Again, in the last, I wanted to also mention that the primary function of the small subunit is decoding. Right? What the codon in the mRNA should be decoded so that the adapter molecule, and it's a process basically. It's not like in what you see it as computer or encryption and so on. In large subunit, the primary function is peptidyl transfer because it has the peptidyl transferase center. That is where the polymerization of amino acids is happening, or the production of the protein is happening there. So based on the decoding, the sequence is getting converted into the sequence of proteins. Okay, so the ribosome, the first component to bind to the mRNA is the small subunit. The small subunit binds by a sequence that is complementary to the SD sequence that is present on the mRNA. The 16S rRNA is of the small subunit. And here you have a start codon and there is a stop codon, spacer. If the spacer is too long, then the efficiency of the translation initiation could be lesser. Okay, so the spacer, the start codon, and as I said, the SD sequence, this is the consensus sequence, AGG. But in different upstream of different open reading frames, you may have little bit variations of this SD sequence. This is the optimal SD sequence, that is the highest efficiency with which translation could initiate on that particular mRNA. So, and we also spoke about initiator tRNA. It is special. It is loaded with with an amino acid that is formulated N-formyl methionine, right? N you say it because the formyl group is on the amino group, and that is the first amino acid that will be added to. I mean, this is the initiator tRNA that is special, which initiates the translation. So basically, the mRNA is recognized by the small subunit. And I think I also should tell you about these sites in the ribosome: A site, P site, and E site. So both these are kind of imaginary in a way, but that is where the reactions happen. What I meant is they are overlapping or they are kind of fused. MNSL site, this is peptidyl site, and this is the exit site, A, P, E. As we will see in the next slides, and if you have already watched the animation, you would appreciate that the tRNAs will come here, except the first one. The tRNAs will come here, then they will move into the, as the ribosome will move this way. So the tRNA will come to the peptidyl center, and then eventually to the exit center, and the tRNA will be left out. And so you can also see it another way is amino acid will come here, okay, along with the tRNA. And in this way, here it is amino acid is there, that is along with the tRNA. In P site, you have it as a peptide because this is where the protein is growing, new protein is forming. That is peptide site. And here in the E, you kind of have tRNA, but there is no amino acid to it. You can also think of it as empty tRNA, just saying. Okay, so I hope you will be able to remember what is A site, P site, and E site. Amino site, peptidyl site, and exit site. Okay, now mRNA is recognized by the small subunit, and it comes along with the initiator tRNA, and that initiator tRNA is already present in the P site. Okay, this variation is only during initiation. Whatever I spoke just now about here, that tRNA comes here, then moves to the peptidyl site, then to the exit site, and then out of the ribosome. But the initiator tRNA, it already occupies the P site. This is the P site in the small subunit. This would have been the A site, and here E. It already occupies the P site, that is where it is. So ribosome binding site could be somewhere around here, and a ribosome is bound, and the initiator tRNA is, subunit is bound, small subunit. Initiator tRNA is present along with the charged, with the amino acid N-formyl methionine, and then the second, the large subunit is arranged on top of it. Okay, and that's how initiation happens. So you can break it into recruitment of ribosome, the charged initiator tRNA. You have to remember that charged initiator tRNA in the P site, precise positioning of the ribosome on the start codon. So this is the ribosome binding site, and here is the start codon. That is why spacer is important. If the spacer is too long, say if the ribosome binding site was somewhere around here, and the spacer is somewhere else, then it is then the start codon is somewhere else, it would decrease the efficiency of initiation. And I think you can remember this. mRNA recruitment is by base pairing. Where does base pairing occur? This is the place between this region of 16S rRNA and the SD codon. That is how the ribosomal subunit binds to the mRNA. This is with prokaryotes. So initiator tRNA, we spoke of this N-formyl methionine. And there are factors that are involved. And before going, majority of the proteins would have actually had this N-formyl methionine. Okay, N is for the amino group, this is the formyl group, and you have a methionine. Methionine is a sulfur containing amino acid. So once it is formed, the protein is formed, the formyl group can be removed by deformalize, or the initial some amino acids of the protein could be broken up by aminopeptidases. Okay, what we are going to see now is about, we'll talk about the many other factors that are involved in translation. Okay, as a concept, what is happening to the information is one thing, right? The sequence of nucleic acid, based on the sequence of nucleotides in mRNA, the ribosome is producing a sequence of amino acid, which is going to form a protein, a functional protein or a structural protein. And that is information wise if you're viewing it. When you see it as a mechanistic view is how things are happening. That is what the focus is now. There is also chemistry of it. Chemistry view is how is the tRNA, the codon anticodon interaction happening, and how is peptidyl transferase reaction happening, or charging of amino acids to tRNA synthetase by tRNA synthetases. How is it happening? That is the biochemistry way of seeing it. And then you have the mechanistic way, how things are happening. Just now, what we described in initiation is one of the things, that binding, and then so on. Then for this mechanistic way of understanding, we need to remember several factors, such as, in this case, those are called as initiation factors. So they are simply put as IF, initiation factors. And there are three important initiation factors in prokaryotes. First one, IF1, is it occupies, I hope you remember that this is the A site, this would be the P site, and this one is the E site here. In the P site, we already have the initiator. This is representing the tRNA, initiator tRNA like this with the F-met tRNA. So the job of IF1 is to prevent any tRNA binding here. Okay, any other tRNA, or it's actually, it is occupying A site, so nothing else binds there. And then we have IF2. IF2 is the one that facilitates the binding of the initiator tRNA to the P site, and it is a GTPase. And we will just remember GTPase, it will consume a GTP, the energy released by hydrolyzing a GTP to GDP. So it is an energy consuming one. And then we have IF3. So as we were discussing before, the mRNA is something like this, and we said the small subunit first occupies here. The small subunit has a certain degree of interaction with the large subunit. Okay, that's why they are also able to interact. If IF3 is not present, the ribosome like this will bind up like this. Instead, if IF3 is present, right, if it is occupying like this, the large subunit will not be able to bind to this. So in prokaryotes, initiation typically occurs as separate subunits of ribosome. Small subunit recognizes the mRNA, and then the initiation complex is organized on top of it. A fully organized ribosome like this cannot initiate translation. So that means these should be as separate subunits. They should not become fused. And to keep them separate, the job of, that is what is done by the job of, it is the job of IF3, initiation factor 3. So let's look as a sequence of events now. Here are ribosomal subunits, 50S and 30S. It can be as a 70S subunit, or they can be separate entities like 50S and 30S subunits free. So they can bind and unbind, right? It's a kind of dynamics. But translation will be initiated by the smaller subunit only. So the binding of IF3, as we discussed in the last slide, once if IF3 binds, then the 30s and 50s subunits will remain separate. And that is when we have IF1 binding in the A site, and we have the IF2 along with GTP binds to IF1. The binding of IF2 will facilitate the binding of the tRNA at the P site. So E site is occupied by IF3, A site will be occupied by IF1, P site is occupied by the initiator tRNA. Okay? And then from here, the first thing that happens is the IF3 is released out. Once the IF3 is released out, what can occur? This reaction is favored. That is binding or formation of a complete ribosome. So the large subunit will bind and form will form a complete ribosome along with the mRNA. But initiation does not start yet. A site is occupied by initiation factor 1, and initiation factor 2 is not required because its job of bringing the tRNA, initiator tRNA, is done. So once this complex is organized, the initiation factor 2 will hydrolyze the bound GTP and will release as GDP and PI. And then there will be a conformational change, that is the structural changes, because of which IF1 and IF2 are released out. Now what we have here is mRNA, a complete ribosome with the small subunit and the large subunit, along with the initiator tRNA in the P site. That is the initiation complex. Okay. Yes, that is the end of initiation. Now what should progress is elongation. But what we will look before going further is about the initiation in eukaryotes. First, I would want to compare the same factors because it will be easier that way. Prokaryotes and eukaryotes. I hope you are able to view it. So you have IF1. The job of it is to block the A site, and in eukaryotes, it is called as the similar factor is called as IF1A. In somehow, I mean, as you can understand from the complexity level, prokaryotes are simple. They're fast, efficient, and simple. Eukaryotes are little complex and sophisticated. So IF1, and here in eukaryote, you call it as EIF1. E is for the eukaryote initiation factor 1. And there are other factors there'll be, so you call it as IF1A. You may have IF1B and so on also. But we are not going to discuss in entirety because translation itself can be one whole book if you consider regulation and so on. The job of IF2 is to facilitate initiator tRNA binding at the P site. And in eukaryotes, you call it as EIF2. And there is another factor which also plays a role in doing the same is EIF5B. And IF3 is preventing ribosomal subunit association, formation of a complete ribosome. The same function is done by EIF3 in eukaryotes. But there is a significant variation at the level of initiation, and that's to prepare the mRNA and also a little bit about the ribosome as well for initiation of translation in eukaryotes. For that, we need so many factors: EIF1, EIF4A, and so on. I will get back to this slide in next class again for reviewing, for what do you say, for review. But right now, I will just finish the mechanism. Okay. Yeah, let's see. It'll get little complex. You can break it down into pieces, and we will see how we can make a small um, story out of it for remembering. So this is the the ribosome, large and small, APE sites, and then we have EIF1. EIF1A is the one that occupies the A site. I hope you remember that. There are, when I was saying this, you have preparation of ribosomes that is shown in A, and preparation of mRNA that is shown in B. So I'm going to come one after one. First, we'll talk about ribosome, preparation of the ribosome for initiation of translation, because it is the same as what we have learned. It is almost similar to what we have learned in the previous one, in prokaryotic translational initiation. So EIF1A, and we have EIF5 and EIF3 is here. It's a little larger here. So they have these functions. They bind to the small subunit like this. Then EIF2, along with GTP. EIF2, like IF2, is a GTPase. So it will, when it is bound to GTP, it has a different conformation, and it binds to the tRNA initiator tRNA, and then it brings it facilitates the binding of the initiator tRNA at the P site. Okay, that is the preparation of the ribosome, ribosomal small subunit. You call that as 43S pre-initiation complex. Okay, and now we will look at the preparation of the mRNA. I hope I have emphasized a lot on the GCAP, on the 5' that is present on the 5' of eukaryotic mRNAs. And the start codon is somewhere very far away, little bit, if not very far away. And there will be the Cossack sequence, or the ribosome binding site, is not as important or kind of as influential as in the case of prokaryotic initiation. Here, there are multiple components that will actually become essential for initiation of translation in eukaryotes. The GCAP is recognized by IFE, initiation factor E, and then we have EIF4G and EIF4A. They interact. The recognizing one is E, and to the E, you will have the binding of G and A. And then there is the for binding of EIF4B, right? So what is happening is the mRNA could be because of base pairing, they might form hairpin loops like this, if there is certain degree of complementarity. And these proteins act something like helicase. They will unwind the mRNA, and they will keep on moving until, they will unwind the mRNA. We'll stop there, and we'll go to the part where these two will come together. This is the part. One second. Okay, so we have the 43S pre-initiation complex formed here, and then we have the mRNA bound recognized based on the GCAP. Remember, if this GCAP is not present, then you will not have the binding of E to it, and then this mRNA will never get translated. Instead, it might get degraded by the RNases. So the 43S pre-initiation complex binds to the mRNA like this. So the proteins that are binding to the mRNA have several functions. One is to recognize the mRNA, and the second point is to bring the bring the pre-initiation complex to the place of mRNA. So I hope you can you can recognize how different the initiation between difference in the initiation between prokaryotes and eukaryotes is, right? So now we have a kind of initiation complex that is formed here, where we have the mRNA, the small ribosomal subunit, along with the initiator tRNA. But initiation is not complete because we need the large subunit too. So the EIF2, so the ribosome that is formed here, ribosomal subunit, will scan this mRNA. So this is where the start codon is present. Okay? And the ribosome will keep on moving forward while it is still attached to the tip along with the E and other factors. So basically the mRNA is pulled inside until the start codon comes to the P site. And there you will have the base pairing of the initiator anticodon loop of the initiator tRNA with that of the start codon. And that is where it stops. And when that interaction happens between codon and anticodon here, in this place, then you will have changes. First change is, one of the important changes is the activation of GTPase activity of EIF2. Then EIF2 will hydrolyze the GTP to GDP and PI, and it will lose affinity for the initiator tRNA and other factors around, and all these things will fall off. EIF2, EIF1, EIF5, EIF4B, and so on. And there is something else that will take over that is called as EIF5B. Along with it is also a GTPase, it binds there. And then we will have the 60S ribosomal subunit coming and binding on top of this. And once that is done, you'll have the hydrolysis of the GTP by EIF5B, and then it changes its conformation and comes out as EIF1A. I mean, also EIF1A is removed, and now you have complete initiation complex. So the A site is free to allow more charged tRNAs to come. There, every other factors is removed except maybe the factors, some things that accessory factors that are there. Okay. So if I have to make a notes for this, how I should make is, you don't have to draw the diagrams for your notes sake. When you're drawing it as notes, you should know what is EIF1. I usually put what is the way that is depicted here, right? Small subunit, what are all the binding to this small subunit? If I'm writing here, then I can just put it as 40S, then EIF1, right? Then what else is coming? EIF5, 1A, and 3. We know the functions of each of them, if not all. At least EIF1 we know, occupying A site, 3, preventing the association of 60S subunit to the 40S, and 5, and so on. That will be useful which will bind to the E site. And then you will bring, after that, I'll put another arrow, and I will put it as EIF2 along with GTP. GTP is always the active form in a way. And then you can also write as initiator tRNA. When you're writing here, I think it is written as methionine. In eukaryotes, I don't think there is N-formyl methionine. I think N-formyl methionine is for in prokaryotes. And then you will have the 43S pre-initiation complex. So if I can write this little more clearly, and that is what I can have a look at before the exam so that I can remember the order of things. The same way you can write for the mRNA as well. You will write as E. Of course, one should know what is the function of each of them. First one is EIF4E, I think. Then That is the one that recognizes the GCAP. That allows the binding of AF4G and 4A. And then you will have the binding of 4B. And then we will have the binding formation of this complex. So writing in some kind of code or very simplistic way, which can stimulate your stimulate your ability to recall the fact, is how you should want to remember. It's like making a map or a small cheat sheet so that you can recall the things from there on. Okay, so there is it is also thought in eukaryotes especially, there is circularization of the mRNA. It is a polysome, like we have discussed in the prokaryotic in the beginning of this lecture. This is GCAP, and at the end of the 3' end has a poly-A tail. So it is thought that there is an interaction between the factors IF initiation factors G with that of poly-A binding proteins. So basically it is complete, it's like a circle now and continuous. Initiation starts here, and then ribosomes will keep moving along the mRNA, and stop codon termination will occur, and the ribosomes will dissociate. And again, a new set of recognition will happen. And this is a continuous rounds of translation happens. Translation, elongation, termination, and again, reinitiation. So this adds, first thing it will do is it is beneficial to the mRNA. It will not get degraded very fast. Anytime you have mRNA like this, is much more prone to degradation compared to something that is occupied in it like this, right? Basically, it's a kind of accessibility issues. Secondly, it is nice way of recycling the ribosomal subunits. They might terminate here, and they are spatially arranged very close to the start codon where initiation should occur, and then they are going to make up new ones. That's also a possibility. So I will, there are, typically we said something about eukaryotes. They're all monocistronic. That means they have one start codon and they have a stop codon, right? A start and a stop. And each mRNA is supposedly has only one open reading frame. But in some cases, rare, it's not a general phenomenon, but there are some things that one should understand. And why we are discussing it now is it helps in initiation of translation. So we are talking about something called as UORFs, upstream open reading frames. So you see this mRNA, you have a start codon here and the stop codon. So this is one continuous open reading frame. But there is also another another small open reading frame here. This is the second open reading frame that is downstream, is the actual one that produces a functional or structural protein. The function of the upstream, the product of the upstream open reading frame is actually not so much. There is no, there is not as much of the things, a product of the function to these proteins. It is thought that what happens is when the ribosome is getting translated, when the ribosome is translating, ribosome initiates here, and then it translates the small open reading frame. And once it comes here, what should happen? Termination should happen because you have a stop codon. And then the 60S subunit will separate, and 40S subunit will separate. What is thought is sometimes what may happen is a new initiator tRNA might bind in the P site, while the small subunit is still attached to the mRNA. And then Even rarely, it might travel for a while, and on very rare occasions, it might reach to the start codon here. And here again, a reinitiation would occur, and then translation would have happened of the downstream gene. Okay. So I will give you, I will repeat this once again, but I want you to find out the rationale for why is that required, right? There should be some reason for it. You can read the textbook, it's in the box. Try to read, and there are several things in this chapter in the box about antibiotics and also about transfer tmRNA, transfer messenger RNA. Those are some of the concepts that are fantastic to learn, and I would want you to explore this particular box in, I can't remember box number and all, but try to read about upstream open reading frames. Okay, regulatory purposes. So I'm repeating this. You have an open reading frame. That's the actual protein that produces a functional or structural protein. And in some cases, they have a small open reading frame that is upstream of the gene that we are talking about. So upstream of the ORF that is functional. So you call it as UORF. They're small. They produce a small peptide. So translation would have initiated there and then would have terminated at the stop codon. And the subunits would normally be dissociated. And when dissociating, the 40S subunit, the small subunit, could still be attached for a brief moment of time. During that time, on rare occasions, okay, when I say rare occasions, say for example, if 10 times this happened, termination, maybe 1 or 2 times, you may have a tRNA binding to the P site, reinitiation happening, and then formation of and then it might rarely, even rarely, it might reach the start codon, that is downstream, and then an initiation complex may form, and then translation of the second open reading frame may happen. So try to find out what is the functional role of this kind of arrangement. It seems like stupid, right? Why do you want all these things? Why not have directly the open reading frame that we are talking about, or that is important? One thing you should understand is in evolution, there is nothing called perfection. There is nothing called the most logical way. In evolution works by this way, by principle. Whatever works is fine. At the end, whether that particular system or rationale or the reaction or a structure, you can take anything. If they are helping the organism survive, then they will be selected. It doesn't have to be the smartest or the greatest solution. Whatever works is fine. But again, it is all in relation to the degree of selection. I mean, I would still encourage you all to have a look at have a look at evolution, especially because it's important for our ability to think clearly and think in when there is uncertainty, what would happen. And it's called as evolutionary thinking. So I would encourage you all to have a look at that. Try to understand what is evolution, and don't reserve or don't stick to the lines or principles of what you read from the textbook. You can apply it to anything. Evolutionary thinking is applicable to almost every field. It is about how you see how change happens, and how one change in the environment will select for changes in the entities that exist in that environment. Everything will change, and trying to study how this change has come about, or try to know about what kind of changes may happen in the future. That's everything is uncertain, and trying to reason those things is what will bring about your ability to reason. So try to do that. Read more of evolution. And I will also touch upon this one slide, and then I will discuss the elongation in the later slides, next class. Okay, I'll just give a brief introduction. In the next class, I will continue again. So there is something called. There are also other variation of initiation, like UORF is one variation to initiation, right, in eukaryotes, and there is another one. It's called as IRES, internal ribosome entry site. Typically, in initiation, what we have seen is ribosome enters through the 5' cap in a way, right? E binds here, then G, and so on. And then you have the small subunit coming. It is entering from the tip 5' side. So what you say about internal ribosome entry site is something like you have a ribosome. Where is it? When there are sequences apparently that are present somewhere, and using those sequences, ribosome can possibly form in there. That is the meaning of the word internal ribosome entry site. It is thought it one of it. I like this structure here. I don't know where am I writing. Yes, sorry. Here, so the mRNA is thought to fold into certain structure. And to that structure, you have the binding of EIFG. Okay. Here, what you're seeing like a knot-like structure is the mRNA is folding into a structure. It's not the tip of it, but this folding happens at the in there, and then that is allowing the binding of EIF4G and so on, and then the 43S complex forms and the pre-initiation complex, and eventually you will have the binding of the large subunit, and translation is happening. In this case, the recognition is not happening based on GCAP. In the other initiation that we have discussed in the previous, not the URFs, but prior to that, we learned about initiation starting with GCAP, the recognition of the mRNA by the presence of GCAP. Here it is not GCAP. It is a structure, something that is formed by the ribosome. Sorry, the mRNA. Those are called as internal ribosome entry sites. Yes, there is also another one. You can see it here. It does not require all these initiation, not so many initiation factors. There is another kind. If you notice it in a three-dimensional here, of course, it is a schematic. It folds into a structure that is similar to that of a tRNA. And that tRNA fits between somewhere around in the P site, E site. So the ribosome, a ribosome can bind to it like this, because the structure is already resembling that of a tRNA. It fits into the gaps between the 40S and 60S subunit in the PE sites, and then there you will have the rest of the ribosomes, tRNAs coming in through A site and translational initiation. It's like directly going into elongation phase, right? These are some of the variations. I will not ask you, well, we will discuss this in the next class. But remember initiation, then remember that there are variations to initiation like URFs, and there are also IRES. I gave you a work to read about UORFs and what is the significance? Why should it be there? Why not? What is the probable logic of it? Okay. Next class, we will learn about elongation and termination. Hopefully, and I would also suggest you to have a look at something I will